Hi everyone, this is Carrie. Fiveable. Happy Sunday evening. Hopefully you guys have had great weekends and are feeling refreshed and rejuvenated for the school week coming up. Maybe a fall break coming up soon or something fun. So we're going to get started tonight. Hopefully you've been tuning in. We've been going through all the topics for units 1 and 2. We're about to wrap up with unit 2, but more on that in a second. But we're going through the progression of topics that College Board puts out from Unit 1 through Unit 8. So hopefully we're hitting some topics that are related to what you're talking about in class. And if not, just know that we will cover everything eventually. And you can always sign up for Fiveable Plus to watch our replays, especially when it gets to be May and you're really, really stressed about that big exam coming up, which seems like it's in the far off future, but it'll be here before you know it. So that's what we're here for, obviously. Tonight, our topic is facilitated diffusion. So we're going to switch over in a second. But facilitated diffusion in and of itself is definitely a very important topic, but it's kind of combining a lot of topics. So if you have been tuning in, we'll do a brief review of the plasma membrane because that's something that's going to be really, really important when it comes to facilitated diffusion. And then I'm also going to preview a little bit with tonicity, so like hyper, isotonic solutions, because that involves facilitated diffusion. And I'll also do a little preview of action potential in our neurons, which you won't talk about probably for a while. Most teachers leave that as one of the later units. So lots of good things coming up. So remember to follow us on all of our lovely social media. They're doing a lot of promotions lately with like free t-shirts and all sorts of fun things. So definitely get on those social media platforms to follow us. As I said, lots of good things coming soon. So tomorrow night is tonicity and osmoregulation with Jessica. And then we'll have mechanisms of transport, cell compartmentalization, and then the origins of cell compartmentalization. And then by the end of October, so in the next 2 weeks here, we'll actually review Unit 2. So that's all things cell and cell structure. So if you remember Unit 1, chemistry, now we're moving into cells. And then up next, we're going to talk more about cellular energy and all the important things that go along with that, like enzymes, cellular respiration, photosynthesis. So that'll be our next unit, Unit 3. But that's what's coming up as far as our next 5. What am I trying to think of? Oh, streams for you guys. And then, as I said, so we'll do a review of the plasma membrane. We'll talk a little bit about concentration gradients tonight. We'll talk about what facilitated diffusion is, obviously, because that's the name of the stream. And then I'll give you 2 pretty specific examples, and those will be for aquaporins and then sodium and potassium channels. And both of those things, if you haven't learned about them already, you will. So it's a really good preview where you'll be a little bit ahead of the game, and you'll actually get to see why facilitated diffusion is important, because it's very simple for me to explain what it is, but it's a little bit more interesting and also more important that you know why it's important in our bodies and in the scheme of biology. We know with the AP exam, they love to kind of marry a bunch of topics together to make sure that you have a really cohesive picture and understanding of all of these really, really important things. So that's something to keep in mind. I'll always try to weave in a couple different units just so that you can see how they all kind of interconnect, because that's how they show it to you on the exam. Okay, so the plasma membrane. Sorry, I probably shouldn't have put this on like a gray slide, but this is what our plasma membrane looks like. So hopefully you tuned in. I think it was Jessica. I can't remember who talked about the plasma membrane. It might have been Kari, but really, really important structure, has a lot going on. You can see that there's a lot of moving pieces here. It's a very complex part of our cells. I would argue that it's the most important part of our cells. So you'll see there's a lot of proteins. Those red globs that you see are a lot of proteins embedded in the membrane. We're going to be talking about a lot of those proteins today, specifically with protein channels. And then you can also see glycolipids, phospholipids. So a glycolipid is going to be a lipid, which we know is in the fat family, okay, that's attached to a sugar. So anytime you see glycolysis, which we'll talk about next unit, glycogen, which is going to be stored sugar, glycoprotein, which you'll also see, all of that means is sugar. So sugar attached to, in this case, a lipid. If it was a glycoprotein, like we see actually right here, this red guy with the little green chain off top, that would be a glycoprotein. All of these things are going to help the cell in some way. They'll deal with cell communication. They'll deal with cell transport, which is obviously the focus of tonight. They'll communicate with other cells. We have special markers on our plasma membrane that tell our cells that we are who we are, which is why if you get a blood transfusion and it's the wrong blood type, or if you get a tissue like a heart transplant and it's not been pre-screened rigorously enough, your body will attack it like it would any other foreign thing because it doesn't recognize you as you. And all of those markers exist in the plasma membrane. So it does a lot of things. It's incredibly important. And the actual mechanisms behind the plasma membrane are what make it super specific for transport, which again, I mean, review of the plasma membrane just because it's so important. But what we really care about tonight is transport within this lovely structure. So here's where I want to focus for a second. And again, I know that if you, access denied. Sorry, I just got distracted because I see that there's access denied to the slides. That's interesting. Let me post the link and let me know if that helps. And if it doesn't, I might need to change the editing settings. But Qing Long, let me know if that works for you. Still denied. Okay, give me 1 second. Let me just change my sharing settings. Change. Cool. Okay, let me know now. Perfect. Awesome. I must have forgotten to change my sharing settings. Thanks for letting me know, guys. Sorry about that. I definitely want to make sure that you can follow along on the slides yourself. Okay. So as I was kind of mentioning, again, you may have heard this, you may have attended a different session, but it's always good to review these things. And honestly, there are so many questions on that exam that have to do with something with chemistry and the plasma membrane that it's always worth talking about. So what you'll notice here, I like to think of them as like little wiggly men, okay? That is a phospholipid, and we call it a bilayer because there's 2 layers of them. And you'll notice that there's 2 distinct parts. We have a hydrophilic head. So the part that's red that's pointed to, it says hydrophilic head. That is one, water loving. That's what hydrophilic means, lover of water. But it's a phosphate group. So that's where the phospho of phospholipid comes from. Phosphates are polar molecules. That means that they have an unequal distribution of charge, okay? And it means they're going to be attracted to other polar or charged molecules. So water is a polar molecule. Water and the phospholipid and the phosphate heads, they get along great. And that is important because the entire interior and exterior of our cell is an aqueous solution. So this was actually, was it this past year? I think so. 2019, it was a free response question all about the plasma membrane. I should actually post that for you guys. I will in a second. Or we can take a look at it if we have time at the end. But so that's that part. So we have aqueous inside, we have aqueous outside, and then nicely enough, we have these phosphate groups that love water that are oriented towards the aqueous solution, okay? Then in the inside, protected from the water, are these hydrophobic tails. So hydrophobic, water-fearing, and these are fatty acids or lipid tails. So we know oil and water, they don't mix. Lipids are incredibly nonpolar, okay? They have really strong carbon-hydrogen bonds that are incredibly nonpolar. They are not water-loving. They do not want to be near charged things, okay? So they're oriented towards the inside for that reason. Now, the most important thing and the thing that we will talk about a lot today is that these fatty acid tails, I like to call them, and I come up with a lot of weird analogies if you've watched my streams in the past. I like to call them like the Kraken tails, if you're familiar with like the mythical sea monster, the Kraken, because they just kind of look like tentacles. These tentacles, these hydrophobic tails, they are really the deciders of who gets to and doesn't get through the plasma membrane. Okay, so sometimes students get confused because they're like, oh, the phosphate group is water loving, so water should have no problem getting through the plasma membrane. It's like water has no problem like being inside and outside of the cell, but water's not going to be able to get through those kraken tails, right? Those tentacles that you see in the middle. Okay. And so they're really the deciders of who does and doesn't get through the membrane in which particular way. Okay. So that's just an important thing to know. Any questions about the structure of this? I'm sure you guys have seen it by now. You may have seen it in a previous stream. Any of those things. Okay, cool. Again, just good review, and I'll show you at the end the free response question that was all about the plasma membrane. Okay, so who gets through the plasma membrane? So we kind of already talked about this. So the molecules that are going to have absolutely no problem getting through the plasma membrane by themselves, no help necessary on their own, are going to be, and you can see it in the upper left-hand corner here, small uncharged molecules. Okay. Reason being uncharged, right? Usually we would say nonpolar, but uncharged somewhat similar, although I don't love that because water is uncharged. But I like to say small nonpolar molecules are the ones who are able to get through the plasma membrane easily. Why? Because they're small, right? And again, this is like, think of this as a barrier, right? Smaller things can get through more easily. But two, because they're able to get past the tentacles, right, who are also nonpolar, right? Like is going to love like here. And so if something's nonpolar, it's going to be able to kind of coast through those nonpolar fatty acid tails, no problem. So really good examples of things that can go through the plasma membrane through what we would call simple diffusion, meaning like no energy required. That's what any diffusion would be like carbon dioxide and oxygen. They're small. They're nonpolar, they share their charge very nicely, and they can scoot through the plasma membrane very, very easily, which obviously benefits us because we are constantly bringing in oxygen and releasing our carbon dioxide that we've produced as a byproduct of cellular respiration. So that works really well for us, and that's all well and good. But obviously not all things are small and uncharged, okay? So that's what we're kind of talking about today. So as I said, small uncharged, and then with the gradient is the last important thing here. So if we take a look back at this picture, so I don't know how familiar you guys are with concentration gradients, but they're relatively straightforward. So a concentration gradient just means, so gradient just means change, right? It means that we have two areas that are different than one another. A concentration gradient means we have two areas of different concentrations. So for example, on the left-hand side of this image over here, we have a very high concentration of whatever these blue molecules are on this side of the cell membrane, and we have a very low concentration of those blue molecules because we have none on this side of the cell membrane. Okay, so high concentration, low concentration. The change in those concentration values is called the concentration gradient. Okay. Molecules, and this is essential to know if you haven't heard it already, molecules will always, 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 always move from high concentration to low concentration, okay? And you can see that happening here. The molecules that were outside start to come inside until eventually, over time, if they're able to, they will reach equilibrium, which is where we have equal number of molecules inside and outside. That is the happiest place that a molecule can be, where everything is equal and we don't really have a concentration gradient anymore because we don't have a change in concentration. Okay, so I like to think of this a couple ways if you haven't heard it before. So one, if you picture concentration gradient as a mountain, if you're going from low, the bottom of the mountain, up to high, the top of the mountain, that takes energy, right? Molecules are not going to naturally go from low to high or climb up the mountain, okay? And I'll show you an example of that sometime during this presentation. But if you're going from a high concentration at the top of the mountain to a low concentration at the bottom, that will happen naturally, right? You will roll down the mountain naturally. No energy required, just a few bumps and bruises, okay? So going from high to low requires no energy. Going from low to high will require energy. So that's an easy way to remember it. And just remember, everything likes to spread out. Nothing likes to be crowded together, just like humans don't like to be crowded together. So that's how things are naturally going to flow. Okay, so now that we know that for something to go through simple diffusion, they're small, uncharged, and with the gradient, we're going to talk about how all those other things get through the membrane and then specifically focus on facilitated diffusion. So here are your types of transport. Simple diffusion, I just talked about, it's passive, which means no energy required, okay? And it's for small nonpolar molecules, and they need to be going with the concentration gradient. So again, from high to low. Facilitated diffusion, our focus for today, when we eventually get there, is also passive, meaning that no energy is required. And these are for small polar molecules with the concentration gradient. So they're still going from high to low. We're still not using any energy, but this time our molecules are polar, and therefore they need a little extra help to get through those tentacles or fatty acids in the inside of the plasma membrane. Active transport, you may have heard of before. Active always means energy. Like, think about it. If you are active, you are using energy, okay? So active transport is used for bulky molecules, meaning like larger molecules who can't just squeeze through the plasma membrane on their own, and for molecules traveling against the concentration gradient. And I'm going to show you an example of that as well, a really important example. I think the most important example. But sometimes we are going to force things against the gradient because it allows us to do other things, and more on that in a little bit. And then lastly, endo and exocytosis. So endocytosis is bringing in either something large or a large quantity of something from outside of the cell. A good example would be like a white blood cell engulfing a bacteria cell. And then exocytosis would be releasing from the cell into the outer area a large molecule or large quantities of something. So this is sometimes used if a cell is sending out like massive amounts of a cell signal. They'll use exocytosis. And that for the most part will use energy. Okay, any questions about that? You know, there's an ask a question button, or you can also type in the chat box, whatever is easier for you guys. But I would be more than happy to answer any questions. Is glucose considered large? That's a great question. Glucose transport, let me see if I can find a picture. Transport into the cell. They actually are another example of facilitated diffusion. So they can get through without the use of energy. Yeah. Oh, yeah. Okay. So it's called GLUT is what they call it. It's a glucose transporter, and they will go through no problem, unless for whatever reason the cell has. So that would be facilitated. Yep. So they use a transport protein, but as long as they're traveling from high to low, which they usually are, because you usually would have more glucose in your bloodstream than inside of your cell where they're consistently breaking it down, that would be facilitated. The only time that could potentially be active is if you had a high concentration of glucose in your cells. But otherwise, glucose will naturally travel into your cells through the glucose transporter. And actually, some like glucose regulation problems actually occur with that glucose transport protein. But I have a picture similar that I can show you in a second. Great question. Okay. Okay, so facilitated diffusion in its essence, again, is very simple, which is kind of why I'm talking a lot about other types of transport in comparison to this one. And I'm also going to give you some examples, because really all it is like it's diffusion. So diffusion, if you can remember that that's just the movement of molecules from high concentration to low concentration. This is just that process, but with a facilitator, right? So your teachers facilitate your learning. They're guiding you. They're helping you get there. This protein is a facilitator. It is helping the molecules to transport to where they need to be. So what you'll notice with this blue protein right here is that it's completely preventing the interaction of whatever this green molecule is with the fatty acid tails, right? It's almost like a little bead or like a little hole punch through the membrane that's completely coated, usually with molecules that are going to attract or chemically attract, and I can show you that in a second, whatever molecule they're trying to bring in. okay? But those molecules do not have to interact at all with the fatty acid tails, okay? So that's all facilitated diffusion is. Still, the molecules are moving from a high concentration to low concentration. It's not active transport, even though a protein is involved, a transport protein is involved. Still passive, still no energy. Molecules are moving from high to low. We just now have a tunnel, basically, to get past those fatty acid tails. So that's really all it's about. So not too much going on with that, but I have some good examples that show you why this is so important. So any questions about the process itself? Okay. So as I already mentioned, small, charged, and with the gradient are your rules for facilitated diffusion. Okay, again, still going with the gradient. Sometimes students think that once a protein gets involved, energy is required, and that is not true here. Sometimes it's true if you have active transport, but facilitated diffusion does not require one. Okay, so some common examples of facilitated diffusion. One of the very most important ones is the movement of water, right? We need water for everything. We're constantly maintaining water balance. So if you tune in tomorrow night with Jessica, she will talk about tonicity and osmoregulation, which is the regulation of the amount of water inside and outside of our cells. But water is polar, so water cannot get through the plasma membrane on its own. So water actually has this lovely channel, which we call an aquaporin. So aqua, water, porin, like a pore, a water pore. So that makes sense. Pore meaning hole, not like poor or like something like that. And so you'll notice that this channel is actually positively charged, right? Which is going to kind of draw the relatively negative oxygens through it. And it's going to allow water, again, to move with its concentration gradient because water is not being forced in or out of the cell. It will just move in the direction of its concentration gradient from high to low. Okay, so that's an aquaporin, and that's what leads to situations that we'll see on the next slide, which again, I'm just going to talk about very briefly because we'll talk way more about it tomorrow night. But when water moves out, so if there's low concentration of water outside of your cells, if you're like really, really overly hydrated, maybe, low concentration of water. If you're really, really heavily dehydrated. And so you actually have more water in your cells than you do outside of your cells. We call that hypovolemia, meaning you have like low blood volume. You don't have very much water in your blood. Your cells can actually start to shrink because the water, which is in a higher concentration within the cell, will start to leave the cell. And you can see this shriveling here, and we call that hypertonic. So I always think if you're hyper, you run outside. So water is running out of the cell from high to low concentration into your the rest of your bloodstream, like your plasma. And you can see those cells shrinking up. Isotonic is really, really happy. Iso means equal. So you can see that water is moving in and out here. So we would have equal concentrations of water inside and outside of our cells. So a really classic example here, if you've ever had an IV before, the IV fluid is 0.9% saline or salt, and that's because your cells have about 0.9% solute inside of them. So if we just used pure water, we would be like in the situation on the right where there's more water outside of your cells than inside of your cells, and we'd actually be at risk of the water entering your cells and then bursting. So instead, we give you a solution that's equivalent to the solute in your blood. So those are just some examples. So aquaporins are going to be the facilitated diffusion transport protein for water in order to create all of these three scenarios. And again, this is just blood cells, but it would be in any cell. And again, lots more on that tomorrow. Okay, so then the other really, really important topic to talk about are sodium and potassium channels. So again, you probably haven't talked about an action potential yet in your classes because they're very complex, and you won't talk about them until you talk about the nervous system. But they are like the second most or the most, I don't know, depends on how you want to think about it, important use of diffusion, facilitated diffusion in particular. So an action potential takes place in your neurons, your brain cells, right? Your nerves. And so these happen every time that your nerve sends a message. Anytime your neuron sends a signal to another neuron. Okay, so me moving my arm, the fact that my heart is beating right now is because my brain is sending an impulse to the the SA node of my heart and having it pump. So all of those things constantly happening use these channels. So what you need to know, because we always need to know about the concentration gradient in our different scenarios, and you can see this here, is that our neurons, I call them salty bananas. Okay, and I call them that because potassium, this red molecule, is inside of our neurons when our neurons are at rest. This is our resting state. Okay, so that's the banana because potassium is in bananas. And then sodium, like salt, sodium chloride, is on the outside. So the banana is here, and then outside is the salt. So it's a salty banana, okay? Potassium inside, sodium outside. When an impulse happens, like you have a thought or you're ready to move or you need to breathe or pump your heart, a couple things happen. And I'm not going to go into all of the specifics just because you don't need to know them probably quite yet. But the first thing that actually happens when an impulse is being ready to be sent is that sodium channels open up. So really quick about sodium. Sodium, Na+, it lives in a positive state, right? Because it is in our group on the periodic table. It has one single valence electron in its outer shell, okay? And it wants to lose that single valence electron so it can have the full octet underneath and be a lot more stable. So it is a positive molecule, which means it's polar and therefore can't get through the membrane on its own. It can only get through the membrane with a transport protein, okay? Because of that, we can actually create some sort of concentration gradient. Same with potassium. Potassium is also positive, K+. Okay, so both of these things cannot get through the membrane unless they're going through their specific channel protein. So first thing that happens when you're sending an impulse is we have sodium channels, which is this green one. They open up, and you'll notice that sodium goes from high concentration to low concentration inside of the cell, and our cell starts to get really positive. We say it gets depolarized, but more on that when we talk about this later. Okay, but the cell starts to get really positive because we're adding more positive ions, right? Sodium is positive. Okay, as that happens, this gets sent down. We eventually need to start to equalize our cells again because we're not continually sending that same message. So this message of positivity will kind of get sent down the cell, and then the cell needs to start to reset itself. So the first thing that it'll do is it'll close these pumps so that no more sodium can come in, and it will open up potassium pumps. And again, potassium is the same. It's positive, so it can only get through the membrane with a protein channel. So both of these are examples of facilitated diffusion, keeps us alive every day, okay? And this potassium will leave the cell, okay, through the potassium channel, because again, from high concentration inside of the cell to low concentration outside of the cell. Okay, so potassium moves out and the cell starts to get more negative. Okay, so that's all well and good for the time being. We've kind of started to reset the cell, but in order to fully reset the cell, we need to get it back to salty banana status. We needed to get it back to where potassium is inside and sodium is outside. Okay, and that involves this lovely pump that is viewed in pink here called the sodium-potassium pump, which you may have heard of before. It is probably the most important pump that we have in our body, and it is an example of active transport, meaning that it is going to use energy, and it's one of our largest users of energy because we're constantly resetting our neurons. If you think about how much we are moving, doing, thinking, breathing, all that kind of stuff. So what sodium-potassium pump does is it forces things back against their concentration gradient. So what's not truly shown in this picture is even though it looks like we don't have any sodium outside of the cell anymore, we still have a very high concentration of sodium outside of the cell, right? Sodium's concentration in our blood is like 100 times what our sodium concentration is in our cells, okay? So there is still a very high concentration of sodium outside. There's still a very high concentration of potassium inside, okay? So in order to push these sodium molecules back outside where all the other sodium molecules are hanging out, and in order to push these potassium molecules back inside where all the potassium molecules are hanging out, we're going to need to use some energy. We're actually going against the concentration gradient. So it's the opposite of what we had with the sodium and the potassium pump, where we were going with the concentration gradient and therefore didn't need energy. And this pump is going to need to use some ATP in order to reset our salty banana. Sodium on the outside, potassium on the inside. Okay. So that is a very brief explanation of how our nerves work. This action potential is incredibly important and will definitely be talked about later. But it's a really great example of the various types of transport and why they're used. If you think about it, if sodium and potassium weren't charged, right, we would not be able to form a concentration gradient like this, meaning we wouldn't be able to have this action potential because they just go through the membrane any way they would like to, just like oxygen and carbon dioxide do, right? They'll just diffuse naturally. But because they can only get through with those particular channels, we actually have a lot of control because those channels, if they're closed, no sodium's getting into the cell. If they're open, that sodium will be able to come through. Any questions about those two examples with aquaporins and osmosis and then the sodium and potassium channels? I know it's a lot of information, but facilitated diffusion in and of itself is usually pretty straightforward. Okay, cool. So I'm going to show you then really quick the example. My face is nice and big again. 2019. Okay. I'm just going to pull this up really quick. And I'm also going to share it with you guys. Hopefully you know. If you don't, you can Google any year's free response questions, and they'll have the questions and the answers. It's two separate PDFs. They'll have the questions on one PDF and then the rubric on another. But I would say it's never too early to start familiarizing yourself with the types of questions that have come in years past. Anything going up until 2013. Anything before 2013 is super outdated because that's when they redesigned the exam the first time. You will notice that there's 8 free response questions in this packet, and you are very, very lucky because you only have 6 free responses. This is the first year that has happened, but you still have the same amount of time to answer them. So you are all set. Okay, so here we go. Oh my goodness, wait, it's not in this one. Where is it? Was it in the practice exam? Jeez. Okay, I led you astray. Give me 1 second. Let me try to find it really quick. AP Biology, plasma membrane. 2017, really? I must be losing my mind. This one's a decent example, so we can go over it, but it's not the one I was thinking of, so I apologize. I really thought it was in 2019, but sometimes my years all blur together. Okay, let me pull this one up real quick for us. And if I find the other one that I'm thinking of, it may have been in a practice exam. I'll send it out to you guys. Okay, but this one is another good practice. So it says, so this was question 8. So this is a short free response question. It's worth 3 points. You will have a couple of short free response questions that are good practice. And this one says, I'll make it bigger. Estrogens, so we know about estrogen, it's a hormone, are small hydrophobic lipid hormones that promote cell division and the development of reproductive structures in mammals. So we've heard of this. We know that it's mainly produced in females. And it says estrogens passively diffuse across the plasma membrane and bind to their receptor proteins in the cytoplasm of target cells. So your first point comes from describing one characteristic of the plasma membrane that allows estrogens to passively cross the membrane. So we talked about this. So going back into the question, we see that estrogens are small, they're hydrophobic, meaning that they're nonpolar, and they're lipids. And we know that the deciders of who gets through the cell are the lipid tails. So there's 2 things that you could have put here to get a point. One is the hydrophobic or nonpolar nature of the estrogen, or the other is that there's some space between the phospholipids. So that's something that I didn't focus on as much. But are the phospholipid tails, the fatty acid tails, are kinked. They're unsaturated, not saturated fats. And so there's actually some wiggle room in between there where some small molecules can scoot through. But the big one that probably most students wrote about was that they were hydrophobic. And notice that that's really all you had to write to get that point. They always encourage that you write in full sentences, but you could have just written it is hydrophobic, period. Whereas probably a lot of students expanded upon that a lot, being like, it's hydrophobic, which means blah, 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 blah. But you really just needed to give one characteristic, okay? So that's something good to know. Usually being concise, being shorter with your words is better. If you write more, it often tends to mean that you don't fully know what you're writing about. Okay, so that's 1 point. That's great. And this was the last question on the exam. So usually results are a little bit low for this one. Okay, so then the second part, in a laboratory experiment, a researcher generates antibodies that bind to purified estrogen receptors extracted from cells. The researchers use the antibodies in an attempt to treat estrogen-dependent cancers, but find that the treatment is ineffective. Explain the ineffectiveness of the antibodies for treating estrogen-dependent cancers. So even if you weren't quite sure, and at this point you would have had some immune system and you would have known that antibodies are unable to enter the cell, even if you weren't quite sure, if you knew that it was ineffective and if you knew that the antibodies were binding to the receptors, which it says up top are inside of the cell, bind to their receptor proteins in the cytoplasm of the cell, you can just assume or make a good educated guess that the antibodies aren't able to get to it in practice. They're only able to get to it when it's extracted. Okay, so saying that the antibodies are unable to enter the cell would be a great point that you can get kind of from deductive reasoning. And then your second point comes from saying that antibodies will not bind to intracellular receptors because they're extracellular. And that's something that we'll talk about even more when we talk about with cell signaling. But you usually are either an extracellular or intracellular receptor. And if you can't get through the plasma membrane, you're not going to be able to bind to a receptor that's inside. So that's just kind of interesting example of where some of the things that we're talking about would show up. And I'll always try to show you free response questions just because practice makes perfect. And the rubrics on these things can sometimes be beasts. Like when you do and don't get points can be kind of confusing. And so it's just good to practice with that kind of stuff to make sure that you feel confident when you take your actual exam. Because again, even though it seems far away, it could be here before you know it. Okay, so there's 2 years of free response questions that you can look through and just see what you know, see all that you're going to learn, and just kind of take a peek. But again, the answers are all out there as well. You just need to Google like rubric or scoring guideline or answers, any of those things, then it'll pop up. And you can also search like AP Biology free response cells, right? And they'll pop up with some hits. What I will say is, especially for long questions, those are 10 points. They usually incorporate multiple units. So you may not know all of the information yet if you're, still pretty much in the beginning of your year here in October. So don't panic too much about that, but it's still good to kind of see some real life practice because it's, when it's game time, you want to practice with things that are just as rigorous and challenging as what you're going to see on the real deal. Okay. Any other questions before we end things tonight? Hopefully you guys feel a little bit more confident. Again, facilitated diffusion is pretty straightforward once you feel comfortable with the plasma membrane. So if you've been, tuning in and keeping up with us, hopefully that didn't feel too bad. It's more just review. And again, a little preview of what we'll talk about tomorrow night and then what we'll talk about in a few months to come with the nervous system. Okay. Great. Well, have a great night. Have a great day at school tomorrow. Tune in tomorrow night for tonicity, hard words, and osmoregulation with Jessica. It should be a great session. And then we'll be back. So thank you, everyone. Thanks for coming and spending your Sunday night with me. I'll see you guys soon.